Hello everyone, welcome to a new episode of Biology. This is Mitosis and Cytokinesis. Our key concept for today's lesson is cells divide during mitosis and cytokinesis. The main ideas: number one, chromosomes condense at the start of mitosis. Number two, mitosis and cytokinesis produce two genetically identical daughter cells. These are the new cells that the cell obtain after cell division. So let's start. Chromosomes condense at the start of mitosis. DNA is a double-stranded molecule, as we know, made of four different subunits called nucleotides. A chromosome is one long continuous thread of DNA that consists of numerous genes, along with regulatory information. Your body cells have 46 chromosomes. Can you imagine if these 46 chromosomes are stretched out straight and laid end to end? The DNA in just one of your cells would be about 3 meters long. So, have you ever wondered how these 3 meters long DNA strand fit inside our microscopic cells in each cell? How does it fit inside the nucleus of a microscopic cell? Let's see. DNA wraps around the proteins that help organize and condense the DNA itself. During interphase or when a cell is not dividing, DNA is loosely organized and it looks a bit like spaghetti. This is in the normal case, in the normal part of the cell cycle during interphase, which is gap 1, synthesis, and gap 2. During mitosis, however, the chromosomes are tightly condensed like the one in the picture. These changes in the DNA's organization allows a cell to carry out its necessary functions. During the interphase, proteins must access specific genes for a cell to make a specific proteins or to copy the entire DNA sequence. During mitosis, the duplicated chromosomes must condense to be divided between the two new nuclei equally. So, if the chromosomes remain stringy or thread-like, do you remember when they are loose or like spaghetti? If the chromosomes remained in the same way during mitosis, some problems might happen. They could become entangled, twisted, or crisscrossed. Perhaps a cell would get two copies of the same chromosome or get no copies of a different one. The key to this process is the association between DNA and the proteins. The next figure will show the process that converts a chromosome from a linear strand of DNA into tightly condensed form. So do not be surprised or afraid. So this is the chromosome, this is the double helix of the DNA. Each continuous double-stranded DNA molecules makes one chromosome. Then these chromosomes started to coil or wrap tightly around the proteins at equal intervals, as you can see. So the wrapping around the proteins, it forms chromatin. These proteins are named histones. So the chromosomes wrap around the proteins called histones to form chromatin. Then the chromatins, the interactions between parts of these histones, further compacted the DNA. Then it gives us the supercoiled DNA. It says the chromatin coils more and more tightly around organizing proteins. Then we move to, or we get the condensed duplicated chromosome like this one to your right hand side. The condensed duplicated chromosome can be aligned and separated during mitosis. So this is the one that can be aligned and separated during mitosis. So this condensing and coiling around the proteins to make it easier for chromosomes to be separated during mitosis. Almost all times during the cell cycle, each of your chromosomes is associated with a group of proteins, which is named histones. DNA wraps around the histones at regular intervals, small two bits on a string. Parts of the histones interact with each other, further compacting the DNA. We are seeking the compacting of DNA to make it easier to separate it during mitosis. At this stage, the spaghetti stage, which is a loose combination of DNA and proteins, they are called chromatin. The word loose itself describes how much of the DNA strand folds back on itself. It does not mean the DNA is loosely wrapped around the histones. No, it's tightly wrapped around the histones. As a cell progresses into mitosis, chromatin further condenses, more and more condensation. So, it continues to coil more and more tightly around the organizing proteins, finally forming small, thick rods. Recall that each chromosome has already been copied during the S stage, the synthesis. Yes, so the chromosomes looks similar to an X letter in which the left and the right halves are two identical DNA double helixes. One half of the duplicated chromosome is called the A chromatid. So each chromosome consists of two chromatids, but together the two identical chromatids they are called sister chromatids. Look at this picture here. So this is one condensed chromosome consists of two chromatids. They are identical to each other, so they are named sister chromatids. Sister chromatids are held together at the centromere, the part, a region of the condensed chromosome that looks like it is pinched. So both the sister chromatids, they are linked or held together from this part. In addition, the ends of the DNA molecules form structures called the telomeres, which are made of repeating nucleotides that do not form genes. So these are the edges of each chromatid. They are named telomeres. The telomeres are made of repeating nucleotides that do not form genes. These telomeres prevent the ends of chromosomes from accidentally attaching each other, and they help prevent the loss of genes because they do not have genes, and these are ones to be lost without genes. A short section of nucleotides is lost from a new DNA molecule each time it is copied. So it is important that these nucleotides are lost from the telomere, not from the genes themselves. So the telomeres, they are made to be lost safely during the process of copying the DNA. Then we come to mitosis and cytokinesis, which produce two genetically identical daughter cells. The combined process of mitosis and cytokinesis produce two genetically identical daughter cells. The nucleus and the chromosomes go through dramatic changes in the cell division. So this is a cell, this is the nucleus. Keep in your mind that we have centrioles and spindle fibers. These are centrioles and centrosome. This is the parent cell. Okay, then interphase. Interphase plays an important role in preparing the cell to divide. As you can see, interphase, which is gap 1 synthesis and the gap 2. It plays an important role to for the cell to divide, to prepare the cell for division. It provides a critical time for the duplication of organelles and for DNA replication. By the end of the interphase, an individual cell has two full sets of DNA or chromosomes and is large enough to divide. Then the cell copies its DNA and grows in preparation for division. The DNA is loosely organized during the interphase. Then we come to mitosis. This is the fourth and the last stage of the cell cycle. Mitosis divides a cell's nucleus into two genetically identical nuclei, each with its own single full set of DNA. This process occurs in all of your body cells except those that form eggs and sperm and prepares them for cytokinesis. Although mitosis and cytokinesis are continuous processes, scientists have divided them into phases to make them easier to understand and discuss. Let's see. So the four main phases of mitosis are prophase, metaphase, anaphase, and telophase. Pro, meta, ana, telophase. Cytokinesis begins during the late anaphase or telophase. So let's see, what about prophase? The first stage of mitosis is prophase. During prophase, chromatin condenses into tightly coiled chromosomes. Okay, each chromosome consists of two identical sister chromatids, as we said, the X letter. The nuclear envelope breaks down, the nucleolus disappears, and the centrosome and centrioles begin to migrate to opposite sides of the cell. Organized microtubules called spindle fibers grow from the centrioles and radiate toward the center of the cell. So this is what happens during prophase. DNA and the proteins condense into tightly coiled chromosomes. As you can see, these are the tightly coiled chromosomes. The nuclear envelope breaks down. The centrioles begin to move to the opposite poles of the cell, and the spindle fibers form. Then the second stage of mitosis, which is metaphase. In metaphase, the spindle fibers attach to a protein structure in the centromere of each chromosome. So the spindle fibers attach to part of the centromere or a protein structure in the centromere from each chromosome and align the chromosomes at the cell's equator. Align the chromosomes at the center or in the middle of the cell, as you can see around the middle of the cell. Okay, then the third stage of mitosis, anaphase. Now, during anaphase, the sister chromatids separate from each other. As you see, the sister chromatids, they are separated from each other. The spindle fibers begin to shorten, pulling the sister chromatids away from each other and toward the opposite sides of the cell. Then telophase. In telophase, a complete set of identical chromosomes is positioned at each pole of the cell. As you can see, the nuclear membranes started to reform again around the new nucleus. The chromosomes begin to uncoil and the spindle fibers fall apart. There is no need for them again. Then we move to or get to cytokinesis. Cytokinesis usually begins in late anaphase or telophase. Cytokinesis divides the cell's cytoplasm into two new cells and completes a full stage of the cell cycle. Cytokinesis differ in animals and plant cells. In animal cells, the membrane forms a furrow or a trench that is pulled inward by tiny filaments like a drawstring, like this one. So part of the cytoplasm is pulled down like a trench. Gradually, the membrane pinches closed, forming a separate cell around each nucleus. This is in case of animal cells. But what about in plant cells? During cytokinesis in plant cells, the membrane cannot pinch inward because the plant cells have a hard, rigid structure named the cell wall. Instead, a cell plate forms between the two nuclei, a cell plate. It is made by the Golgi apparatus, which supplies the new plasma membrane. Then a new cell wall or a new wall that grows as cellulose and other materials are let down. Typically, cytoplasm is divided evenly between daughter cells in both plant and animal cells. The formation of new cells is critical in both multicellular and single-celled organisms. The single-celled organisms use cell division to reproduce, not to build more cells, whereas multicellular organism uses it for growth, development, and repair. Hope you have enjoyed the episode. See you in the next one. Stay safe and study hard. Thank you guys and goodbye.